Hello everyone and welcome back to Biology with Mrs. Evans. So in the last video we talked about mitosis, the division of somatic cells. In today's video we're going to be talking about the division of sex cells, or gametes as we call them. So the process is similar in the fact that it has the same 4 stages, but it's different in the fact that mitosis has 1 nuclear division and meiosis is going to have 2 nuclear divisions. So meiosis has 2 nuclear divisions in which the cell is going to divide its nucleus twice. So you're going to end up with 4 haploid cells, because remember, haploid is basically the half the number of chromosomes. So because the nucleus divided twice, you're going to end up with 4 cells that have half the number of chromosomes that the normal diploid parent would have. So difference between mitosis and meiosis. Mitosis, remember, is identical cells. So it starts off with 16, it ends in 16. And then meiosis is basically the creation of 4 genetically different. We're going to talk about how that happens in a minute. But they are going to start off at 16, but at the end result of meiosis, you're going to end up with 8 chromosomes basically in each cell. So N stands for the number of chromosomes in an organism. So for humans, what is N for us? Remember, we have 46 chromosomes in our somatic cells, so N for us would be 23, and for 23. And then our gametes would have 23 chromosomes, because haploid means basically half or 1 set of chromosomes. So meiosis again has 2 nuclear divisions. So the first division we refer to as meiosis I. The second division is called meiosis II, and the end result again is you're going to end up with 4 cells that have half the original amount of genetic material. So you're going to end up with 4 haploid cells. So first division of meiosis looks like this. So again, you have interphase before meiosis, the first division of meiosis, because again you need the cells to divide, so you need to copy DNA. After interphase, you have the same phases that you see in mitosis. You have prophase 1, metaphase 1, anaphase 1, and telophase 1. And you'll notice the difference between mitosis and meiosis. Basically, if you look at the chromosomes here, so in metaphase 1, the homologous chromosomes basically are going to line up. So you have blue would be your mom's, and then orange would be, let's say, your dad's chromosome that you got from your dad. So they line up next to each other, and then in anaphase 1, the homologous chromosomes themselves are going to split apart. So the stages are similar. The difference is that in anaphase 1, the homologous chromosomes are going to separate. So the same things happen in prophase 1. Chromosomes start to condense, they become visible, the nuclear envelope is going to break down, the mitotic spindle, as you can see, is going to form in prophase 1, and then metaphase 1, just like in mitosis, the chromosomes are going to line up in the middle. The difference between anaphase in meiosis and anaphase in mitosis, remember, in mitosis, the sister chromatids are going to separate. So in anaphase 1, the homologous chromosomes are separating. And then in telophase, it's similar to what you see in mitosis. The nuclear membrane is going to reform basically around the nucleus, and the DNA will condense. So meiosis 1. So sister chromatids, all right, we talked about those. Those are what makes up basically the chromosomes. And then in anaphase 1, you'll notice that the homologous chromosomes are going to separate. What you'll notice on this picture is that a small little piece of, let's say the blue is your dad's chromosome, has broken off and swap places with a small piece on your mom's chromosome. And you'll see the same thing happen with these. So this is called crossing over. When chromosomes are so close to each other, pieces of your mom's chromosome can break off and basically trade places with pieces of your dad's chromosome. As a result, this creates 4 genetically different cells that are created. And this happens during basically anaphase 1 of meiosis, when the chromosomes are close together. It can also happen in metaphase technically as well, because again, they're close to each other and they haven't started to move. So crossing over when homologous chromosomes can exchange basically pieces with one another. So the second division is the second division of the nucleus. This time, notice there is no interphase because DNA has already been replicated in the first interphase, and the goal is to end up with cells that have half the genetic material. So if it replicated now, that goal would not be achieved. So there is no interphase in between the first division and the second division. So again, prophase 2, you're used to this. It's the same as you've seen before. The nucleus is going to break down, chromosomes are visible, mitotic spindle forms, and then if you notice, the second division of meiosis looks pretty much the same as what you would see in mitosis. So in metaphase, chromosomes are going to line up in the middle. And just like in anaphase of mitosis, now the sister chromatids are going to separate. And you'll notice that the end of meiosis are 4 cells that are genetically different, because you would have had crossing over that created the differences in the 4 cells. So Meiosis II, again, like I said, looks pretty much the same as mitosis. The only difference is that the cells are going to have half the number of chromosomes, because there is no duplication in between Meiosis I and Meiosis II. So remember that an X-shaped chromosome that you see is a duplicated form of DNA. So here is a picture representation basically of what happens in meiosis. So you start with a diploid cell, and that means it has 2 sets of chromosomes. It undergoes basically the first division, which is going to end up with 2 cells that are still maintaining that diploid number. And then each cell will undergo a second nuclear division, where each cell now is going to have 1 set of chromosomes. So haploid, they're going to be haploid, or half the number of chromosomes. So notice that DNA is going to be replicated basically in interphase, which is the first part of the cell cycle for sex cells. So DNA is going to be replicated so that the end result for the first division is 2 cells that have the same amount of DNA as the original. And then the second division ends up with 4 cells that are haploid. All right, so you need to be able to explain this process. So gametes, the formation of gametes, depending upon whether it's a male or female, has a different term. So for males, the production of sperm cells, as you see here, is called spermatogenesis. And for males, 1 cell is going to divide to create 4 genetically different cells that have everything that they need to be functioning cells. For females, our process is called oogenesis, and that is the formation of an egg cell, or an egg. And then 3, it used to be that we thought they were useless polar bodies, but we now know that polar bodies are going to help basically provide nutrients to the developing egg cell. So it creates 1 completely functioning cell, and then 3 polar bodies that are going to help basically provide nourishment, etc., to the cell. So meiosis summary, again, you're going to end up with 4 cells that are genetically different. They are genetically different because of crossing over. Unicellular organisms do not undergo meiosis, and that is because, remember, they're going to undergo mitosis, which is simply asexual reproduction. They don't have more than one cell in them, so they do not make basically sex cells. So meiosis, 2 nuclear divisions creates 4 genetically different cells because of the process of crossing over. So here is crossing over picture for you. So notice that during it's technically anaphase is when most of it will occur, but it can also happen in metaphase, like I said. During meiosis, the first division of meiosis, the chromosomes are so close to each other that they can basically join together to create a tetrad. And when they do that, they can swap pieces. So if you notice the next picture, you'll notice that part of mom's chromosome ended up on dad's chromosome and vice versa. So this is dad's chromosome, so this is part of mom's ended up on dad's, and part of dad's ended up on mom's chromosome. And that process is called crossing over. So that's when basically a tetrad is formed during the first meiosis division. So genetic diversity, why is it important? Genetic diversity is the key so that no 2 individuals are the same. So by creating these new combination of genes, it allows for organisms to have basically different characteristics so that they can potentially survive an outbreak of disease because we are genetically different. If we were all the same, if a disease would wipe out one, it would wipe out all of us. But by having that diversity, it ensures the possibility that at least one or not a few of us would survive. So again, it's important that we have this genetic diversity to ensure basically the survival of a species. So one thing that you'll need to be able to do is to compare and contrast mitosis and meiosis. This is a version that I have done for you. You're going to want to pause the video and copy this down into your notebook. Again, mitosis creates 2 identical cells because it has 1 nuclear division. So if it started off with 16 chromosomes, it's going to end with 16 chromosomes. Again, it is the division of somatic cells, so all the cells that are not our sex cells. And so for humans, it's going to create cells that have 46 chromosomes in them. And then meiosis, remember, is the division of sex cells or division of gametes. And it undergoes 2 nuclear divisions. So it's going to create 4 genetically different cells. And they are genetically different because of crossing over. Remember, crossing over occurs during the first division of meiosis, in particular the anaphase. It can happen as well as in metaphase, but typically we say anaphase. And again, 2 nuclear divisions. So what can we say for the similarities? We can say that both of them have interphase that precede them. All right, because remember, DNA has to be replicated, so interphase, I'm just going to abbreviate that. It is both of them create new cells. All right, so create new cells. And that should be an A up there. All right, create new cells. They're both followed by cytokinesis. They both contain genetic information, etc. So be able to compare and contrast mitosis versus meiosis as well. And that is basically cell division in a nutshell. So we've talked about division of somatic cells, we've talked about the division of sex cells, and we've talked about the whole process itself is the cell cycle, and you'll need to know basically be able to define what the cell cycle is, which remember is the stages that a cell goes through in its lifetime, and then be able to tell me the stages that the cell goes through. So interphase broken down into the 3 parts: G1, S, G2, the M phase, which is the mitosis phase or the meiosis phase, and then cytokinesis is the division of the cytoplasm. And then once the 2 new cells are created, it basically starts that process over again. So those 2 cells would grow, and then the cycle would continue for those new cells. Again, that's it for cell division. Hope you're having a great day. I will see you in class. Bye.